Hi, I'm Lewis Quayle, data scientist, bioinformatician and maintainer of lewisdoesdata.com, which is my website where I blog data science tutorials and guided data analyses. Welcome to the fourth and final part of this RNA-Seq tutorial series where we're going step by step from raw sequencing reads to publication ready results. In the earlier tutorials, we've processed raw RNA-Seq data, conducted quality control and performed differential expression analysis. So now what we want to focus on is how to visualize these data and use data visualization techniques to summarize and communicate our statistical analysis results effectively. So as with part 3, we're going to be continuing directly on from where the previous tutorial left off. So what that means is that the libraries are going to be the same, the custom plotting theme is going to be the same, and I'm going to set that as my default plotting theme for ggplot2. So I'm just going to run all of those lines of code. So I've also made a ready-to-use R data file downloadable via the blog post that accompanies this tutorial for anybody wanting to just jump in at this stage and do some data visualization. So I'm just going to run that code chunk as well, and now we can see that we've both got the DDS object and the clean results object within our global environment. So volcano plots are great for visualizing differential expression results. They plot the inverse log 10 adjusted p-value or the p-value on the y-axis against the log 2 fold change on the x-axis, highlighting significant genes. So we're going to start with the most simplistic version here. So we can see that this is simply a scatter plot where we've plotted the inverse log 10 p-value on the y-axis against the log 2 fold change on the x-axis. This is a good starting point, but it lacks detail. So for instance, it doesn't distinguish between significant and non-significant genes. So by adding a new column to indicate significance, we can colour the points accordingly and then make the plot more informative. So the first thing that we're going to do is add a variable to the clean differential expression testing result data frame. Let's just run that to remind ourselves how that looks. So this is the data frame where we have all of the results post differential expression testing from the last tutorial. And what we're going to do is we're going to mutate on this variable called signif, and that's going to be a Boolean vector where the observations will evaluate to true if the adjusted p-value is less than 0.05 and the absolute log fold change exceeds 1. So if we run that entire chunk of code, we can see now that we've added this signif variable on the right hand side here. It's of type logical and it contains true where that statement evaluates to true and false where it evaluates to false. And what this then allows us to do is pass another aesthetic mapping into the AES function inside the ggplot2 call, and that will colour the points depending on whether they're true or false. So if we run that line of code now, we can see that we've effectively, what we've done is highlighted those genes that we would deem significant based on the criteria that we've defined. So this improvement does highlight significant genes, but we can take it a step further by distinguishing between the up and down regulated genes. And we can do this inside the mutate call using the case when function to categorize points instead of using a simple Boolean vector to indicate whether they are significant or non-significant. So what we're doing here is saying when the adjusted p-value for one of the genes post differential expression testing is less than 0.05 and the log fold change is greater than 1, label it as an upregulated gene. When the adjusted p-value is less than 0.05 and the log fold change is less than -1, label it as a downregulated gene. And if the adjusted p-value is greater than 0.05, then it's a non-significant result. And then we're creating the same aesthetic mapping by mapping the newly created signif variable onto the color scale, and then we're actually manually specifying that color scale, that discrete color scale, by passing in a vector of character strings, which are the names of colours that are used in R. So if we run that entire block of code, we can now see that instead of having true false for the signif variable, we've now got the downregulated genes highlighted in light steel blue. We've got the non-significant genes here in grey, and then we've got the significant upregulated genes in royal blue. So already, having used those couple of relatively simple measures, we're already increasing the amount of information that this volcano plot's going to convey. Another thing that we can do is label specific genes, and this can enhance the plot's usefulness, especially when highlighting either biologically significant findings or genes which belong to pathways of interest. Instead of overwhelming the plot with all the gene names, we can selectively label, say, the top 10 up and down regulated genes. So that's the first thing that we're going to do. We're going to create a vector of symbols for the upregulated genes by filtering the differential expression testing result, where there's a entry where the log fold change is greater than 0. And then we're going to take, because it's already arranged in order of the adjusted p-value, so the most significant genes are at the top, we can use this slice head function to take the top 10 results. So if we just run that, you'll see that effectively that just cuts the data frame down to the first 10 observations where the log fold change is greater than 0. And then what we can do is we can use this pull function call here to pull out the symbol column as a vector from that data frame. So if we run that, we can now see that we've removed that data frame structure, but we've still got the genes, the gene symbols in the order that they're presented in the table now as a character vector. We can assign that to the up object within the global environment, and then we can do the same thing for down-regulated genes, just changing the direction of the logical test for the log fold change here to be less than 0 instead of greater than 0. So if we save that, what we can do now is we can take the res clean data frame, and we can use, inside the mutate call, we can use the same case when statement to create that signif variable on which we're going to colour the data points, but then we can create a second variable called label. So what we're saying here is if the symbol character string within the symbol vector in the res clean data frame, or the symbol variable within the res clean data frame, if that exists within either the up or down character vectors, then assign the values from the symbol variable. Otherwise, just give us an empty character string or a blank. So if we just run that entire mutate statement now, we can see that we've not only added the signif variable created using the case when statement to indicate the directionality on which we're going to colour, but we've also added this vector of labels that we can then use. So now if we pass that to the ggplot function with the same initial mapping as before, so we've got the log fold change on the x-axis and the inverse log 10 p-value on the y-axis, but now you'll see that we create this global mapping where the label scale is mapped to the label vector that we've created globally, and then we've actually moved the AES call with the color scale mapping to the signif variable down to the just to the geom point layer. So what that means is that our points are still going to get colored, but the labels are not going to get these same colors assigned, and then we've created the same the discrete manual color scale there, and then we're using geom text repel to actually plot those labels. Although really, strictly speaking, I should move that up to there. So if we run that, it might take a little while to run. There we go. We can now see that we've not only created our color mapping that shows us the down-regulated genes and the up-regulated genes, but we've also managed to label the top 10 points with their respective Hugo gene symbols within each of those two groups. So by combining these elements, we can customize the plot further to suit our dataset and biological question. So like I mentioned earlier, instead of highlighting the most significant genes, you might choose to highlight genes in a specific pathway or those related to a particular hypothesis that you have. So you can use these techniques that I've showed here to customize the basic volcano plotting code to meet your specific needs. And in the blog post, I've showed a fully customized version to demonstrate what you might produce by experimenting with ggplot a little further. So now we're going to move on to look at expression plots. So an expression plot is a powerful but underutilized tool for visualizing the expression levels of individual genes across experimental conditions, offering an intuitive way to validate statistical results by showing the actual distribution of normalized counts. So these both directly complement the volcano plot and act as a visual aid to confirm differential expression. To build a basic expression plot, we first need to extract the normalized counts for the genes of interest. So in the following example, I've chosen the top 30 most significant genes, but this could be tailored to highlight, for example, a specific pathway or gene set as well. So what I'm doing here is I'm extracting the 30 most significant genes and assigning that to this top res object. And then we're asking for the normalized counts matrix back relating to from our DDS object, which we have to pass via the estimate size factors function if that hasn't already been done, to ensure that we get the counts that have been normalized with respect to library size back. And then we're going to filter that down to just the genes that are contained within this top res object that we've just created. So that then gives us a counts matrix. So we're just printing the top 10 results for that counts matrix, or the top 10 observations in that counts matrix. But you can see that we've now got a counts matrix, although it is technically a data frame. It's a counts matrix which relates to the top 30 genes within our differential expression testing result. So the next thing to do is to reformat the data into a long format suitable for plotting. And since Tibbles in R don't support row names, we first need to move the row names, which you can see in the output of the previous set of function calls here, to their own variable, which we're going to call ensemble, before we then pass that into the pivot longer function. So if we inspect the result of that, we can see now that what we've done is we've taken that data frame, and having moved the ensemble row names into their own column, we've then pivoted the values for each sample from the wide into the long format. So what we now have is a row for each gene relating to each replicate within each experimental condition and the respective normalized counts value. So we should have, in this tibble, we should have 9 times 30 entries, which is exactly what we've got here. We've got 270 rows, and that's because we've got 9 repeats of each gene name, and that's because there's 3 replicates within each of the experimental conditions within our data set. So at this stage, we can create a basic expression plot, and we'll create a grouping variable from the sample IDs so that we can colour the data points by replicate groups, and also that'll enable exclusion of the TGF group data because this wasn't part of our initial contrast of interest. So that's exactly what we're doing here, and then we just pass that filtered long format data frame into the ggplot function. So you can see now that the expression plot gives us the normalized counts values for each of the replicates within a particular group. And in this case, our test group, our irradiated samples, are coloured this bluey green colour, and our control group are coloured this reddy pink colour. In order to refine the plot a little more, what we can do is we can swap the ensemble IDs for gene symbol annotation, and we can reorder the genes by the significance of differential expression, so the adjusted p-value, and then we can add the metadata for additional annotation, such as colouring by group. So that's exactly what we're going to do here. So if we just print the output of that series of function calls. We can then use this data frame to create a slightly more augmented version of the expression plot. So you can see now we have the Hugo gene symbols instead of the ensemble gene IDs, and we've got the grouping as they appear within the metadata. So just as with volcano plots, that like we created earlier, spending a bit of time refining the plot aesthetics, such as adjusting the colours, axis labels or titles, can transform the preliminary version into a more polished publication ready figure. So you could check out the blog post for an example of how I've elevated this plot a little further. If you had more data points, you could jitter them, or you could consider adding alpha blending, for example, to the individual samples and adding a mean point with standard error or confidence intervals, or you could use something like a box and whisker plot instead of plotting the individual data points. So finally, we're going to move on to clustered heat maps, and these are a powerful way to visualize patterns of gene expression across multiple samples. By clustering genes and samples with similar gene expression profiles, they reveal underlying structure in the data, such as co-regulated genes or the effects of experimental conditions. And this type of visualization is especially useful for identifying broader trends that might not be apparent from other plots. Now, although ggplot2, which regular readers of my blog will know I absolutely love, has a geontile function to make heat maps, specialized packages such as the pheatmap package offer more functionality such as scaling values and clustering the data, and it's all about picking the right tool for the job. So while ggplot2 with its extensions are great for hundreds of plots in R, the dedicated packages out there for making heat maps and related plots like oncoprints are just better for those tasks. So the first thing that we need to do is make sure that we have loaded the pheatmap package, which we should have done at the outset, but I forgot to do that. And then our heat map is going to use variant stabilized counts, as these are more suitable for visualizations when using distance measures, such as in the case of heat maps with hierarchical clustering. And we'll start by selecting the top 50 differentially expressed genes based on significance. So we're going to use a very similar call to the slice min function as we saw when creating the expression plot, just swapping out 30 for 50 there. We're going to save that as this top results object. And because we're pulling out the variant stabilized counts from the DDS object, we're going to use this pipeline that ends in the assay call. And because this is a matrix that we're trying to subset here, we're going to use the base R subset function instead of going through a series of steps in order to be able to filter that as a data frame and then coerce it back to a matrix. This is just quicker. So effectively what we're going to end up with is this top counts matrix, which is the top the counts matrix for all of the samples within our data set for the top 50 genes, top 50 most differentially expressed genes within our differential expression testing result. So with these data we can create a basic heat map. So we're just going to pass in this top counts matrix, and we're going to make an argument that says scale the rows, and we can pass that to the pheatmap function. Now note that the default color palette goes from low expression in blue to high expression in red, which is a good alternative to the traditional red-green heat maps, which aren't suitable for those with red-green color deficiency. And this plot provides a fairly good starting point, but it isn't really very interpretable, particularly due to the ensemble IDs being used as the row labels. So to make it more friendly, we could replace the row labels with the Hugo gene symbols, which is exactly what this call here does. So I'm not going to break it down too much here. There's some information in the blog post about that. But if I print the output again, we can see now that we've still got our top 50 counts matrix, but instead of having the ensemble IDs in the row names, we've now got the Hugo gene symbols instead. And we can pass that matrix to the pheatmap function in the same way that we've just done, and we get essentially the same plot, but now we have the Hugo gene symbols being used as the row names. So another thing that we can do is we can modify the column names to reflect the experimental group and replicate with improved clarity. So that's exactly what this block of code does here. So effectively what we're doing is pulling out the metadata from the DDS object and making sure that that's coerced to a data frame, and then we're selecting the variables that we want from that data frame, and then we're adding a label, a new label variable where we're concatenating the group and replicate separated by some white space. So we can see now that we've created this new label variable here where we've effectively combined the group and replicate columns into one single column, making sure that we're separating them with a space character, and then we're going to pull out that newly created label variable as a vector, so removing this data frame structure and ending in with a character vector. So if we assign that to col labs, we can then pass that vector in to our updated pHeatmap call, and you can see now that the column labels have also been updated to reflect better those experimental conditions and replicate numbers. To add an additional layer of information, we can annotate the columns with sample metadata such as experimental groups using the _col argument within the pHeatmap function. And this can be a little more tricky than our previous modifications, but essentially what this requires is a data frame where the row names match the column names in the matrix passed to the pHeatmap function. So if we just preview that again to remind ourselves, what we need is a data frame where the row names are effectively matching these variable names within the VST transformed counts matrix. So that's exactly what we create here. So we take that metadata, coerce it to a data frame, and then we're renaming the group variable whilst also selecting that. So if we first run the assignment there and then run the updated pHeatmap call, we can now see that we've got coloured annotation bars above the heat map, where our legend here has this capitalized group title, and we can switch that on and off should we wish to. So this annotated heat map offers a much clearer view of the patterns in the data, highlighting how genes and samples cluster together. And this is undoubtedly fit for publication. I've seen far worse in published manuscripts, unfortunately. But for real professional looking publication figures like the one that I've shown in the blog, you can customize the heat map even further. So options include things like modifying the clustering algorithm, scaling color schemes, or even defining the order of rows and columns manually using other algorithms like modular leaf ordering. And doing this relies on a much more in-depth understanding of unsupervised clustering and empirical clustering coefficient calculation, as well as lower level familiarity with the pHeatmap function, which are somewhat outside the scope of this tutorial. That could form a tutorial in and of itself in the future. So the last thing that I wanted to cover here was how to save heat maps while actually hijacking one of the functions from ggplot2. So what we need to do is, once we're happy with the heat map plotting code, to create the heat map that we wish to save, we can add this argument here, silent equals true, which will prevent any output to the plot viewer when we then assign the output to this, in this case, heat map object within our environment. So if we do that, we can see that that's now saved as a list object within the environment, and that then enables us to pass the name of the plot object to the ggsave function, and we can give a file name, specify the device, in this case PNG, making sure that the file extension and the device name match, and then we can specify the path where we want that saved output to go to, and we can also change some of the plotting parameters, so the dimensions of the plotting area, the units that these dimensions are given in, and the DPI or resolution of the output. So with that, we've reached the end of this tutorial and series, and I hope it's provided you with the tools to analyse and visualise your RNA-Seq data confidently. From this point, there are many more analyses and visualizations that you could consider, such as pathway over representation analysis, gene set enrichment analysis or GSEA, graph network analytics such as protein-protein interaction networks, or weighted gene correlation network analysis, for example, or even preparing the data for machine learning applications, which is something that I've considered writing a blog post on as well. Let me know which of these you'd like me to do a tutorial on or series of tutorials on in future in the comments. If you found this helpful, please like and subscribe. Thanks for following along with this series, and I'll catch you again soon. Enjoy RNA-Seq data analysis.